Hi everyone, welcome back. In this video, we are going to look at the overview of adaptive immunity. My goal is not to discuss every detail, but to help you to understand the big picture before we study B cells, T cells, antibodies, and immune responses in greater depth. By the end of this video, you should be able to explain how adaptive immunity develops, how B cells and T cells become activated, and how they work together to eliminate the pathogens. The outlines of this video are 1. Introduction to adaptive immunity, 2. B cells and antibody-mediated immunity, 3. T cells and cell-mediated immunity, and 4. Functions of antibodies and effector T cells. My name is Dr. Mo. I'm Associate Professor from Newcastle University. Our immune system has two major arms. The first is innate immunity, which provides immediate but non-specific protection. It responds within minutes to hours and uses cells such as neutrophils, macrophages, dendritic cells, and natural killer cells. The second is adaptive immunity. Unlike innate immunity, adaptive immunity is highly specific. It usually takes several days to fully develop during the first exposure to a pathogen, but it provides a more targeted and powerful immune response. Adaptive immunity can be divided into two major components: humoral immunity and cell-mediated immunity. Humoral immunity is mediated by B lymphocytes, which differentiate into plasma cells and produce antibodies. These antibodies help to eliminate extracellular pathogens by mechanisms such as neutralization, opsonization, and complement activation. Cell-mediated immunity is mediated by T lymphocytes. CD4 positive helper T cells coordinate immune responses by activating other immune cells, while CD8 positive cytotoxic T cells directly eliminate infected or abnormal cells. Another important feature of adaptive immunity is the development of immunological memory. After the first exposure to a pathogen, memory B cells and memory T cells are generated. These memory cells allow the immune system to respond much faster and more effectively when the same pathogen infects the body again. This immunological memory is the principle behind vaccination. Let's begin with B lymphocytes. B cells develop and mature in the bone marrow. Once mature, they circulate through blood and lymphoid organs as naive B cells. At this stage, they have not yet encountered their specific antigen. When a pathogen enters the body, antigen-presenting cells, especially dendritic cells, activate helper T cells. These activated helper T cells then provide signals to stimulate antigen-specific B cells. After activation, B cells undergo clonal expansion, meaning they rapidly divide to produce many identical cells. Some of these cells differentiate into plasma cells, while others become memory B cells. Plasma cells are antibody-producing factories. They can secrete thousands of antibody molecules every second, helping to eliminate invading microorganisms. Memory B cells survive for many years, sometimes even decades. During a second exposure to the same pathogen, they respond much more rapidly, producing antibodies much faster than during the first infection. Let's now discuss antibodies. There are 5 major classes of immunoglobulins. IgM is usually the first antibody produced during a primary immune response. IgD is mainly found on the surface of naive B cells and functions as part of the B cell receptor. IgG is the most abundant antibody in the blood and tissues. It provides long-term protection and the only antibody class that crosses the placenta to protect the fetus. IgA is the major antibody found on the mucosal surfaces such as the respiratory tract, gastrointestinal tract, saliva, tears, and breast milk. Finally, IgE is involved in allergic reactions and also plays an important role in immunity against parasitic worms. Although these antibody classes have different structures and distributions, they perform several important functions. First, they can neutralize pathogens and toxins by blocking their attachment to the host cells. Second, antibodies can act as opsonins. They coat microorganisms, making them much easier for macrophages and neutrophils to recognize and phagocytose. Third, antibodies can activate the classical complement pathway, resulting in enhanced inflammation, opsonization, and in some cases, direct lysis of bacteria. These 3 functions, neutralization, opsonization, and complement activation, are among the most important mechanisms of antibody-mediated protection. Now let's move on to T lymphocytes. Unlike B cells, T cell precursors originate in the bone marrow but migrate to the thymus where they undergo maturation. During thymic development, T cells rearrange their T cell receptor genes to produce unique antigen receptors. Most mature T cells express an alpha-beta T cell receptor, while a smaller population expresses the gamma-delta T cell receptor. The thymus also performs 2 critical quality control processes. The first is positive selection, which ensures that T cells can recognize self-MHC molecules. The second is negative selection, which removes strongly self-reactive T cells that could otherwise attack the body's own tissues. These processes are essential for establishing self-tolerance and preventing autoimmune diseases. After maturation, naive T cells leave the thymus and circulate throughout secondary lymphoid organs, waiting to encounter their specific antigen. Now let's see how CD4 T cells are activated. When an infection occurs, dendritic cells capture pathogens in peripheral tissues. These dendritic cells then migrate to nearby lymph nodes after leaving the bloodstream through a process known as diapedesis. Within the lymph node, dendritic cells present antigen peptides on MHC molecules to naive T cells. However, antigen recognition alone is not enough. Full T cell activation requires 3 signals. First, recognition of peptide MHC complex by the T cell receptor. Second, co-stimulatory molecules such as CD28 interacting with B7 molecules. Third, cytokines that guide the differentiation of T cells into different functional subsets. Without appropriate co-stimulation, T cells become inactive or tolerant rather than activated. Let's see how CD4 positive T cells differentiate. Once activated, CD4 positive helper T cells differentiate into several specialized subsets. Th1 cells activate macrophages and promote immunity against intracellular pathogens. Th2 cells stimulate eosinophils and B cells and are important for immunity against parasites and allergic responses. Th17 cells recruit neutrophils and help defend against extracellular bacteria and fungi. T follicular helper cells or TFH cells provide essential help to B cells inside germinal centers, promoting antibody production, affinity maturation, and class switching. Finally, regulatory T cells or Treg cells suppress excessive immune responses and maintain immune tolerance, reducing the risk of autoimmune diseases. Each subset produces characteristic cytokines and performs distinct functions within the immune system. The other major T cell population is CD8 positive cytotoxic T cells. Naive CD8 positive T cells, which complete their maturation within the thymus, travel to secondary lymphoid organs such as lymph nodes and the spleen. There, they are activated by antigen-presenting cells with the help of CD4 positive T cells and subsequently differentiate into cytotoxic T lymphocytes, in short CTL, that eliminate infected cells. In the diagram on your right, you can see that naive CD8 positive T cell in the yellow circle is activated by antigen presenting cell in the red circle with the help of CD4 positive T cell in the pink circle. And CD8 positive T cell subsequently differentiate into cytotoxic T lymphocyte or CTL shown in yellow circle. Once they identify an infected cell, CTLs kill it by releasing perforin and granzymes which induce apoptosis. This mechanism is particularly important for eliminating virus-infected cells and many tumor cells. So, in summary, adaptive immunity provides highly specific protection through B cells and T cells and generates long-lasting immunological memory. B cells mediate humoral immunity by providing antibodies that eliminate pathogens through neutralization, opsonization, and complement activation. T cells mediate cell-mediated immunity through CD4 positive helper T cells that coordinate immune responses and CD8 positive cytotoxic T cells that kill infected cells. And finally, adaptive immune responses require effective communication between antigen presenting cells, B cells, and T cells to provide powerful and targeted protection. I hope this video is helpful for you. If you like it, please subscribe and share. Greatly appreciate it. Thank you and have a nice day.